Let's get started. Thank you for joining us today. My name is Carrie Phillips, and I'll be coordinating today's webinar. I'm here with Jackie Carville. Jackie's going to go over how to use DNASTAR's Lasergene Genomics Suite software to analyze exome sequencing data. She'll be using a case study to illustrate how you might look for a causal gene of a rare Mendelian disease. You may have noticed your phone has been muted. However, we encourage you to ask questions. To do this, type it into the chat dialog and send to host. I will then direct the questions to Jackie to be answered for the whole group. All right, with that, I'll turn it over to Jackie. If during the webinar you need any assistance or have any questions, please do send a chat message to me using the chat box.
Awesome. Thanks so much, Carrie. Just to get started here, at DNASTAR, we're a global software company headquartered right here in Madison, Wisconsin. As you can see from the picture here, it's another nice cold day here in January. Our sales team, developers, and support network are all located here. So if you have any questions about our software, we do have the full support staff on hand to answer you, and then use your feedback to convey to the developers and make our software even better for the next release. So to give you an idea of what we're going to be covering today, let's check out our agenda here. So first, I'm going to give some background on DNASTAR as a company, and then we'll have a discussion about our Lasergene Genomics Suite software package. Then I'll go ahead and discuss our Kabuki syndrome case study in some detail before finally getting into a live demonstration of our software. So first, some history on our company. We were born out of the E. coli lab of Dr. Fred Blattner, who was the professor of genetics at the University of Wisconsin in 1984. We built a strong reputation with our early products during the Sanger sequencing years that then continues on today. While still offering these classic sequence assembly and analysis tools, we have expanded into the realms of next-gen sequencing. And here we support all major next-gen sequencing platforms. In addition, we also now offer software to assist in the work of structural biologists. All of our software in these different areas has been developed for research use only. Now, DNASTAR products have long been established on the desktop computer, both on the Windows and Macintosh platforms, but are additionally available on both the Linux cluster or the new DNASTAR cloud. Now, as we can see from this graph, DNASTAR is the leader in citations in peer-reviewed journals. For the past 28 years, more researchers have cited DNASTAR software in these scientific journals than any other sequence analysis software. So the software here is research grade, but it is easy enough for many people to be able to use it, even for some of the most complicated next-gen sequencing workflows. Now, all of these people using our software also have some great things to say. Here are a couple recent comments we've received through Twitter. You can really get in touch with us quickly across all of our social media channels, Facebook, LinkedIn, Twitter, and YouTube, and then let us know how DNASTAR software is working for you. So here at DNASTAR, we offer three main software packages, and our Evolution Suite is what you're looking at here on your screen. And this suite was designed with evolutionary biologists in mind and features tools for performing multiple and pairwise sequence alignments. This suite here also allows you to assemble contigs of Sanger sequences and identify genes, along with including some various basic molecular biology techniques, such as virtual cloning and primer design. Our structural biology suite, as you're looking at in this screen, provides tools for structural biologists to analyze protein sequences, visualize macromolecular structure, predict epitopes, and align structures. In addition, the structural biology suite offers access to NovaFold, which is our new protein structure prediction program. And finally, our Lasergene Genomics Suite features applications for the assembly and analysis of next-gen sequencing data. Now, this is the suite that we're going to be focusing on for the rest of the webinar today. So the core component of our Lasergene Genomics Suite is our SeqMan Engine application. Now, this application will perform reference alignments or de novo assemblies across all major platforms. So whether you're working with Ion Torrent or Illumina data, our assembler does support that. In addition to the SeqMan NGen application, our Genomics Suite also includes the SeqMan Pro and ArrayStar applications for downstream analysis workflows. The variety of applications included in this suite offer the ability for high-capacity, multi-sample analysis. Finally, I do want to point out that our software does come with a dedicated technical support team. You can call or email our technical support representatives or take advantage of our over 200 videos, our training manuals, and our recorded webinars. So one of the most common questions we get from customers performing next-gen projects regards the power and the performance of the software. Now, there are many different assembly options out there, but more often than not, they do need high-power computer hardware setups or Linux cluster options to be able to run properly. Now, our software is designed to run on a more modest desktop computer setup without sacrificing any of these performance variables here. So you can see here some of our benchmarks for our SeqMan NGen assembler. Now, this here is just a subset of our benchmarking data. We do have much more benchmarking information available on our website. But I just want to illustrate here performance across different projects with different data types. So to highlight this chart here, we can assemble the entire human genome, which in this case is over a billion Illumina reads at 36x coverage overnight. So you can set up your assembly before you leave for the day, come back in the morning, and be ready to work with your completed assembly. And here, too, I do want to point out that when I talk about assembly time, this is really the assembly, gapping, statistical SNP calling and filtering, and writing out all of the output files for your project. So it's not just the mapping time, but rather the entire process from pressing start to having a completed BAM file at the end of the assembly. Additionally, our assembler also performs well for de novo assembly options. Coming back to our history as a company, we can very, very quickly assemble this E. coli genome here de novo, taking about 20 minutes to do that. And in fact, 2 peer-reviewed articles found SeqMan NGen to be the best software for performing de novo transcriptome assemblies, earning a #1 ranking for the assembly of both Illumina and 454 data. Now, as I mentioned earlier, traditionally all of our software has been available on a desktop computer. We've designed our software to be able to run on the hardware available to most researchers. And I'll go over some of these hardware configurations in a second. But I did want to mention that in May of 2013, we expanded this traditional software offering up to the DNASTAR cloud. Now, this service allows you to access DNASTAR Lasergene software anywhere with an internet connection. Some advantages to using the DNASTAR cloud offering include access to powerful hardware without having to purchase this hardware yourself, the ability to run multiple jobs simultaneously, the ability to monitor your project from multiple different devices, easy remote collaboration with your peers, as well as some flexible licensing options. So I just want you to keep in mind that the cloud solution is an option for you to run some of these workflows that we'll be discussing today. So if you do choose to run some next-gen workflows on a desktop computer, there are some technical requirements that you'll want to keep in mind. So if you'll be performing some reference-guided assemblies, we do recommend a computer with 16 gigabytes of RAM and a quad-core 3 gigahertz processor. We would also recommend that you have a 1 to 8 terabyte external hard drive. Now, we refer to this as your scratch disk, and this will store temporary files during your assembly run. The size of this depends largely upon the size of your reference sequence used and the amount of sequence data that you're attempting to assemble. So since we're working with human data in this particular webinar, we'll want a larger number of terabytes available on this scratch disk. We would also recommend having a 1 terabyte external hard drive for storing output files from your assembly. So after you're all set up on either the desktop or the cloud, the Lasergene Genomics Suite supports a wide variety of different workflows. Today, we'll be focusing on some of its exome sequencing capabilities, but as you can see, we do offer support for many different data types, assemblies, and alignment workflows. So this webinar today is going to look at the Lasergene Genomics Suite as a whole applied to a very specific case study. Now, this paper, published in Nature Genetics in 2010, examined a Kabuki syndrome. Kabuki syndrome is a disorder with variable phenotype, including a distinctive facial appearance, mild to moderate mental retardation, immunological defects, and other physical anomalies. It is also a very rare disease, with an estimated incidence of 1 in 32,000 births. About 400 cases were reported worldwide since the disorder was characterized in 1981. Now, research suggests this is an autosomal dominant disorder, but in addition, evidence also suggests the syndrome is genetically heterogeneous, potentially arising from mutations in more than one gene. So today we're going to use the exome sequence data from the work conducted in this particular study with our Lasergene Genomics Suite to recapitulate the conclusion that rare mutations in the MLL2 gene cause this disorder. So we'll work through the following software pipeline for the remainder of this webinar. We'll begin by assembling 10 Kabuki syndrome exome datasets and 8 control exome datasets against the human reference genome in SeqMan Engine. We can then examine each of these assemblies in SeqMan Pro before finally conducting our downstream analysis in our ArrayStar application to identify the candidate genes that could be causing this disorder. So with that, I'm going to get out of the software and bring you into the first application that I'd like to discuss today. So what I've opened up here is our DNASTAR Navigator. And this application here is your guide to all of the DNASTAR applications available. So in this hub, you can see a list of all the DNASTAR applications that are available to you, as well as access resources specific to each of these applications. So if I hover over a particular application here, I can see links to videos, tutorials, and FAQs specific to that app. A last nice feature of the Navigator is the ability to access the integrated help system directly from the Navigator. And what's nice about this is it allows you to search across all of the applications. You can also click on any application within the Navigator to directly launch that application. So with that said, I'm going to go ahead and open up the first application in this pipeline, which is our SeqMan Engine application. Now, as I mentioned earlier, this is our assembler application. So I will walk you through how to set up a singular assembly of one of our control exomes in this project. So from this welcome screen here, I'll choose to select Create New Assembly Project. And as you can see, there are a wide variety of different project types hosted within SeqMan Engine. I'll select the Exome Assembly project from the project type window. And since this is our targeted resequencing or exome assembly workflow, we do have the templated option available here in the screen. Next, we can name our project and designate our file locations. Now, here's where you would designate those external disks for the final file location and for the scratch disk that we discussed earlier in the technical requirements slide. But for the sake of demonstration, I'll just choose some local file locations to speed this along. Now, in this screen right here, I'm going to go ahead and add my template file. And you can add files or folders that you might already have on hand. However, I've gone ahead and downloaded a genome template package from the DNASTAR website. So if we go to our website here, we can see that we have many different genome template packages that are available for a variety of different model organisms here. And what's nice about these is that they include the template sequence, annotations, and associated dbSNP, COSMIC, and GURP entries for that particular organism. Again, these are freely available on our website for anyone to download and use. But with that said, I'll go back into SeqMan Engine. I've already downloaded the genome template package, so if I find that file here, I can see that I have that .genome template package file. Open that up and click Next. So here in this screen, I'm prompted to input my sequence files here. So I have unpaired reads in this case, and I do have a folder that's set up with some FASTQ files available. So I'll go ahead and select that folder and import that in. In addition, I do want to select my read technology from the drop-down menu here. And as we can see, we support all of the next-gen sequencing technologies available. This particular exome assembly that I'm doing here features Illumina reads greater than 50 nucleotides, so I'll go ahead and select that. And as I said before, I have unpaired reads for this particular project, but when working with SeqMan Engine, we can definitely support some paired reads as well. One final thing I'd like to point out while I'm in this window is that although what I'm showing right now is just the setup for one assembly. We can also have the option to run multiple assemblies at once. All I would need to do there is load in my separate folders of reads and then select the Run as Separate Projects button here. I'll go ahead and click Next and move on. And now we arrive at our Assembly Options screen. So for most assemblies, the default parameters are going to do just fine. We've been optimizing these default parameters for you throughout the wizard as you make your various selections. However, you do have the option to not only adjust these base assembly parameters, but also to explore some of our advanced assembly options as well. So I'll open up the advanced assembly options here, and we can see that these options include options for the layout, the alignment, as well as SNPs. However, I'm going to go ahead and leave my parameters at their defaults for this particular assembly and click Next. And now we've reached the last page of our SeqMan Engine wizard. We can see that we've created a script for our assembly by going through this wizard process. You also have the option to save your assembly up in this button in the corner here, which is great for when you're running different assemblies or if you just want to have a record of your work. If you have issues with your assembly, you can also send this log to DNASTAR so we can help you to troubleshoot that assembly. Then when I want to begin, I'll just click Assemble to get that started. I won't actually run this assembly here today, but instead I'll now switch into our SeqMan Pro application unless there's any questions.
Hey, thanks, Jackie. Yes, we do have a couple of questions. You said you assembled a folder of FASTQ files for this project. One of our attendees has a FASTA file. Can you use those?
Yeah, definitely. I'll go back to that inputting sequence files screen here in SeqMan Engine. And if I click Add, I can select from this drop-down menu and see the list of supported sequence files that we do allow you to input into the SeqMan Engine wizard.
Great. And how long would an assembly like this take to run?
Sure. So if I'm doing one exome assembly like I'm doing in this particular project, this is going to take about 2 hours to do. If we're going to do many of these together, that does shave a little bit of time off the assembly time. So I know one of our benchmarks is 8 human exomes would take about 14 hours to complete. So, so again, this particular one exome assembly is going to take about 2 hours, maybe a little less. All right, thanks. So I'm going to quit out of SeqMan Engine here and then open up our next application we'll discuss today, our SeqMan Pro application. So our SeqMan Pro application is designed for visualizing our assembly. In this application, I can open each assembly in our project individually and then evaluate them using a variety of views. The screen looks a little complicated right now, but I'll talk about each view individually in a little more detail. So off to the right side of the screen here, I have this nice view, which gives us a list of all of our contigs in our particular project. I can check out the length and the number of sequences in each of these contigs, and I can even choose to sort my contigs by any of these fields up at the top of the screen here. From the screen here, I can also open up each of our contigs into the views I'll be talking about now. So the view that I have open right here is our alignment view, and as you can see, I've opened up this alignment view for our first contig in the project here. And so in this particular view, we're looking at the consensus sequence and the alignment of all of the sequences making up this contig. In addition to the alignment view, we also have the strategy view and looking at the same contig here. And we can use the strategy view to, as I said, further explore all of the contigs in our assembly. So we can use this particular view when we want to analyze coverage in certain areas. We can check the depth of coverage, see different features here from our reference sequence, and we can see a list of constituent sequences here at the bottom section of the view if I expand that down a little more. The last thing I do want to point out in SeqMan Pro with regards to different views is our Snip Report. So I have this open right here. And again, this is for our first contig. So here in the Snip Report, we can view a list of putative SNPs in the assembly. We can also choose to filter SNPs based on the criteria on the top of the screen, such as depth of coverage or impact on different coding features. We can also go ahead and choose to confirm or reject various SNPs as we work through this list. So those are just a few ways that we can visualize and do some basic analysis on each individual assembly. But if you remember, in SeqMan Pro here, we're just looking at one assembly. But our Kabuki project, we're working with 18 total different assemblies. So to do more multiple sample analysis, get more in depth with the project, we'll need to go further into our ArrayStar application. Unless there's any questions on SeqMan Pro here.
Yeah, we do have a couple questions. Is there a way to save the work that you do in the Snip report?
Sure, yeah. If I select this Snip report here, I can easily go File, save the Snip report for that particular config. And what this will do is export out a text file that'll have a list of all of these column headings that you have across the top of the Snip report here. And again, you can choose to remove or rearrange these column headings as desired to really customize this report that you're going to export and save out of the program.
Great, thank you. And what's the best way to search through a SeqMan Pro project?
Sure, especially if you have a really long config, you know, that can take a while, and you want to be able to quickly and easily be able to do so. So I'll show you one option right here. So if I'm in the alignment view and want to search through this alignment and consensus, we can double click on this search term in the bottom corner of the screen here, and this pulls up this search window. And so what we can easily do in this search dialog is to search for SNPs as well as conflicts. And if I do that, I can go ahead and find the next SNP and then be able to quickly and easily scroll through that project looking at the different SNPs that are available. Great. Any other questions? Awesome. So with that, I'm going to go ahead and jump into our ArrayStar application. So before I open up the ArrayStar project specific to our case study, I do want to point out the opening screen of our ArrayStar application. So our ArrayStar application began as a method of analyzing array data. While it still maintains that capability, it has greatly expanded the workflows it supports to encompass projects like RNA-seq data analysis, multi-sample SNP projects, CNV analysis, as well as ChIP-seq and microRNA projects. But I'm going to go ahead and jump into our ArrayStar project for specific to our Kabuki case study. I'll open that up here at the bottom of the screen. So the first view that we're looking at here in ArrayStar is our experiment list view. And here you can see that I've grouped our 18 assemblies into the control samples as well as the Kabuki examples here. From this point, I want to start working on our goal of finding a candidate gene that causes this disorder. Now, we'll do this through a filtering process in which we base our parameters off of prior knowledge about Kabuki syndrome. So to get started with any kind of filtering in ArrayStar, all we need to do is select Filter, Filter All, and then this brings up our filtering window here. So before we filter anything, as some background knowledge, we know that there are approximately 5.7 million SNPs within over 32,000 genes across all of the samples. The first thing we're going to want to do in the filtering process here is to create a SNP subset that we will then use in later filtering. Now, what we're doing here is creating a SNP subset of non-synonymous changes that occur in at least one of the Kabuki samples. The reasoning behind doing this is that causal mutations are expected to be very rare and arise de novo. There are very few recorded cases of familial Kabuki syndrome. Therefore, there would be no reason to suspect that any two assemblies would have the same mutation. So, to create this set, we've opened up this filtering window here, and we'll want to make sure that we're filtering on the SNP level. So what we're going to do first is create a SNP sample group, finding genes in exactly one of the 10 Kabuki assemblies. So to do that, I'm going to type in 1 here, and then make sure to uncheck all of the control assemblies. So then again, we can see that we are doing exactly 1 out of the 10 assemblies there. So we will then want to go choose some additional SNP criteria for this filter here. So as I said before, we're going to want to look for some non-synonymous SNPs to make this SNP set. And we'll also want to change some of the confidence levels here. So I'm going to adjust the p-not-ref score to be greater than 90, the minimum Q-call to be greater than 20, and the minimum depth to be 10. And so after doing that, I can click OK and select Search. And we'll give it a second here to quick search through that 5.7 million SNPs here in our project. So now, yeah, after doing that, what we're left here is this nice list of SNPs that are high confidence, non-synonymous SNPs occurring in exactly one of the Kabuki samples. So again, the logic here is that since the mutations of interest are rare in de novo, each person will have a different variant. So I've already saved these search results here, but for doing any of your filterings, if you do want to save results, all you have to do is select the Remember Results as a SNP set icon, then easily type in your icon, the name for your SNP set, and click OK to save that particular SNP set there. But as I said, I've already saved and created this SNP set that we want to work with for this project. So I'm going to go ahead and move on to our next step in the filtering process. I'm going to go ahead and clear these filtering selections. And this time, now that we've created the SNP set already, we can go ahead and conduct some filtering on our 2 groups of experiments, the Kabuki and the control. So this time, we're going to want to make sure we're filtering on the gene level, because remember, we're looking for those causative genes for this Kabuki syndrome here. And for the first SNP sample group, I'm going to want to find genes with SNPs in at least 7 of the 10 Kabuki experiments here. So again, I'll go through and uncheck controls, and then go back and check that my 7 out of 10 is what I'm looking for. Now we're going to go ahead and select our SNP criteria. And here again, we're going to make a variety of different adjustments due to what we know about this particular disease. So first, we know that the mutation is dominant, so we can make sure that we select any genotype here under the SNP genotype selection. Second, we know the mutation is inactivating, so we will select the nonsense and frame shift options in the SNP options part of the window here. Again, we want to make sure that we have very high confidence levels for these results as we're looking for just that one candidate gene. So I'm going to up the minimum p-not-ref score to 95 and filter on a minimum depth of 10 here. As this is a very, very rare disease, as we've discussed, we know that we assume that it's going to be absent in the dbSNP database, so I'll make sure to check that selector. And finally, we're going to filter on that SNP set that we created earlier. I'll select that from my list of SNP sets and click OK to save those. So now I'm going to go ahead and click this plus icon on the left of the screen to create another level of filtering criteria here. So here in this line of search criteria, we are assuming that the mutation is in the disease samples, but not in the controls. So what I'm going to do is find genes with SNPs in exactly 0 of the 8 control exome assemblies, and then choose our SNP criteria to go ahead and match those that we set for the previous level of filtering. And click OK. And then now I've set all of our filtering criteria. So if we click search here and give it a couple seconds, we can hopefully find the same gene that was left in that paper. And yep, as we can see, we have replicated the result from the Nature Genetics paper in Lasergene Genomics Suite by filtering in ArrayStar, and identifies the mutation in the MLL2 gene as causative of Kabuki syndrome. So now that we've found this causative gene, this really completes our workflow today. But I did want to point out a last couple of interesting features in ArrayStar that you can use to continue doing some downstream analysis on your project. If I exit out of the filtering screen here, ArrayStar has many different integrated views, and the first of which I'd like to point out is our Venn Diagrams view here. So in this particular view, we can compare different types of data. I can adjust the type of data I want to compare up in the top left-hand corner of the screen. This particular Venn diagram I have set up, we're going to compare some different sets of SNPs. So we can compare 2 of these SNP sets or even 3 of these SNP sets using our traditional Venn diagrams view. But in addition to that, we can compare actually many of these SNP sets for analysis by just selecting additional SNP sets and then looking at these in our dartboard view as well. In addition to some of these particular views, we also have a scatter plot, heat map views, as well as gene tables, ways to organize your gene sets or SNP set lists, as well as a SNP table to be able to scroll through your projects and examine all your SNPs. So before I wrap up here for the day, I do want to have a brief discussion on some of our support options that are available, unless there are any other questions on ArrayStar here.
Sure. So are there different advanced filtering options for the different types of projects?
Sure, yeah. So, if we remember when we were in the Filter All screen, we have these 2 different kinds of search criteria for this particular project. We have the ability to search across genes or SNPs here. However, if you're working with different types of projects, you'll see different options for the search criteria in this screen. So, for example, if you're working with some RNA-seq projects, we can search on genes, exons, or isoforms instead of on the genes or SNPs here.
Great, thank you. And just one last question. Is it possible to set up more than just 2 experiments, like the control versus disease in ArrayStar?
Sure, yeah. Our experiment list is pretty flexible here. You can create new experiments quite easily if you're comparing many different groups. In addition, you can create various replicate sets under your experiments here to further organize your options, and just easily click and drag around your different assemblies and things to organize those into however you would like here. So again, you're going to want to use ArrayStar when you're using when you want to compare many different samples with each other. So the experiment list is very flexible to reflect that. Great, thanks. So I'll close out ArrayStar here and then pull up our DNASTAR website so we can talk a little bit about some of our support options. Projects like the one we discussed today can often be a bit tricky to design, especially when figuring out all those different sets of filters that you'd like to create. So I'd like to point out our support options that are available to you. If we go under the Support tab, we have a variety of resources available for your use. The first part I'll point you to is our videos page. Here we have over 200 videos currently on our website in several languages as well. In addition to video resources, under the Support tab again, we also have written tutorials for every application, as well as very detailed online help for each application as well. Additionally, under the Support tab are many other helpful links such as FAQs, technical requirements, archived newsletters, and the option to subscribe if you aren't part of our Lasergene weekly mailing list. and much more. So I'll show you the webinars page where you can access any of our previous webinars, as well as check back for some more scheduled webinars in the future. For the latest updates on DNASTAR, I would also like to encourage you to subscribe to some of our social media channels and our newsletter. In particular, I'd like to point out our YouTube channel. Subscribing to our YouTube channel will give you a first look at many new videos and workflows. In addition, we also now have subtitled videos in a variety of languages to broaden our support to customers around the globe. So that's really all I have to discuss here today. You can refer to our website for more information on all the applications we've talked about. While you're there, I would also encourage you to check out some of the comments from our customers. Our software is easy to use and has an excellent support system available to support your research needs. We also offer fully functional free trials of all the software you've seen today. So I'll take any final questions at this time if there are any.
All right. Well, thank you so much, Jackie. That just about wraps it up for today. Some of the questions we received were more technical than we could address here. So if we weren't able to get to your question, we will follow up with you by email following this webinar. If you think of other questions later, you can always email those to Jackie at webinars@dnastar.com or tweet us on Twitter with our handle @DNAStarInc. And as always, do email us your ideas for future webinar topics. Thank you for joining us. We hope you found this webinar helpful. And as Jackie mentioned, there is a large collection of videos on our website, so I encourage you to check those out when you have time. Our next webinar is coming up on Wednesday, February 26th. In this, Jackie will demonstrate how to create and submit protein structure predictions using the NovaFold application. This will include how to analyze prediction results, and it will include a variety of protein analysis workflows like epitope prediction, structural alignment, and motion visualization. And again, thank you very much for joining us, and we hope you have a great day.